So we'll hand back exams at the end of the day. The O's to the Z's are going to be in the back with the TAs. A's over here, over to N. And as usual, start rustling papers about at 9:45, and then come down when there's space. The key is almost done. I should get it posted later today, so I should be able to get the key up soon, but it's not quite done. Or rather, it's done, but it's really messy, and I need to clean it up. So I want to start by having you describe mammalian kidney function, but I think I want to go through a few points first. I want to get you thinking about why we even need to bother with water balance. Why can't we just tolerate big changes in solute concentrations? Why does it matter? And same thing about wastes. Apart from the fact that they would just take up space, what's the problem with having waste accumulate? So let's talk about that to start with. So in this section, we're really talking about two interrelated factors, water balance and excretion, and then on Friday we're going to talk some more about homeostasis, including temperature regulation in particular. There's two related pieces. One is that the proteins that you have hold a particular conformation, and by now you are highly aware of the fact that the shape matters. In order to have the functions they have to be able to bind properly with binding partners. They have to be able to respond appropriately to neurotransmitters or hormones, and that depends upon their conformation. The conformations they have depend not only on the protein structure, but on the molecules around them. So imagine a part of a protein, so these are all amino acids, and maybe here there's a negative charge, and here there's a positive charge, and those are attracted to each other. Okay? So we have an ionic interaction. And somewhere else, there's a positively charged amino acid, and in a typical cell, that might attract a chloride. Somewhere else is a negative, and that might attract a potassium. Okay, if we change that, if we change the concentrations, let's say we start adding additional sodiums. As we add more and more of them, we might get to a point where there's so many that they don't all get taken up by whatever distribution of negative charges or positive charges there are in the cell. And so we get one of these added sodiums that comes over here. Because it's not attracted anywhere else, what happens to the shape of our protein? Okay, it changes. So one excess sodium, and that's going to make these two parts spread apart, which is what denaturation is. We've denatured the protein. All of your proteins have been selected to have a particular conformation at the solute concentration that's in your body. It's around 300 milliosmoles in a human. So in a human, around 300 milliosmoles, your proteins have the right shape. Get too many sodiums, too many potassiums, too many chlorides, and the shapes start to change. Now, we don't actually know what it is that kills us if we get too far off. But it only has to, for example, mess up the neurons that control your breathing to stop you from breathing and kill you. Okay? It only takes some necessary neuron functioning somewhere to stop functioning right, so that an animal no longer survives. So it is essential that you maintain the right balance, the right concentration between ions and water. It's the concentration that matters mostly. So you could take in more salt as long as you hold on to more water to balance that. It's one of the reasons why a really high salt intake is correlated with high blood pressure. You can tolerate the higher salt as long as you hang on to the water, and so your body, your homeostatic mechanisms are wired. If you have excess salt, either get rid of the salt or hang on to more water, higher pressures. Okay, does that make sense? Okay, the same problem would occur with many waste molecules. So we've already talked about ammonia. And of course, this has the same problem. It could be the ammonium ion, NH+, interacting directly with an amino acid, or it could be that proton isn't held very strongly. It could be that an NH4 happens to be near a protein, the proton pops off, and it gets into the protein somewhere and disrupts its conformation. Okay, does that make sense? So you cannot tolerate high amounts of things like ammonia and ammonium. So one solution to the toxicity of ammonium is to have your nitrogenous waste be less toxic. So here's a urea holding two nitrogens, okay, an NH2 at each end. And that is less toxic. It doesn't tend to pick up that positive charge, but so it is less toxic, but it is polar. The oxygens tend to hold on to electrons a little more tightly. The ends tend to be a little bit positive. So we have weak, it's not an ion, weak, positive and negative charge, unevenly distributed, and too much is also toxic. You can tolerate more urea in your body than you can ammonia and ammonium. Okay, so better, but it still becomes toxic. Too much, and you denature proteins. Same problem. Okay? So you got to get rid of it. That's what we're going to be talking about in the first part of the section today and on Friday. Okay, any questions about this? That makes sense. Okay, there's some terms that are in your notes. There are animals that get rid of their nitrogenous waste primarily as ammonia and ammonium ion, and they're called ammonotelic. Okay, so if it's mainly this, they're ammonotelic in terms of getting rid of their wastes. If it's urea, they're ureatelic. Can't remember if that's supposed to be an O or an A. And then there's another one we're going to talk about. Uric acid, which is what birds, reptiles, and insects do. Ureatelic mammals, many fish, ammonotelic, most invertebrates. Some fish. If you're aquatic, then it's actually pretty easy to get rid of your waste products as ammonia, ammonium ion. Diffuses, NH3 diffuses easily across membranes, so you can just let it go through your gills. And so that's what most animals do. It's cheap and it doesn't build up. Ammonia is in low concentrations in seawater, so you can just let it diffuse out. Not a big deal. Now, there are other wastes you do have to get rid of. So marine invertebrates still have to have ways to get rid of other wastes. But ammonia is not their big problem. If you're a terrestrial animal like us or insects, now you have a problem because ammonia doesn't escape easily as a gas. And so you've got to get rid of it in water, which means you've got to excrete it in some way. And so for terrestrial animals, ureatele and uricatele are more common strategies. Okay, so I mentioned that proteins have evolved, have been selected to match the internal solute concentrations that you have. All the concentrations, extracellular concentrations and intracellular concentrations of the ions and of all the other solutes. Now, seawater has been steadily getting more salty over the billions of years of Earth's history. And there are two strategies for dealing with that, and two strategies in general for dealing with how do you adjust the concentration of your solutes relative to the environment. Osmoconformers, osmo for osmolarity or osmolality. Okay. Osmolarity, to remind you of your chemistry, is concentration of solutes. Osmolality takes into account the fact that when you put salt in, it dissociates into, say, a sodium and a chloride, and so it's number of particles. So we're going to talk about osmoles. Instead of osmolarity, we're going to talk about osmolality, number of particles. Okay. So osmoconformers match body fluid total concentration to the environment. Now, they're still adjusting the ions. They don't have exactly the same concentration of every ion and everything else, but the total solute concentration, meaning that they don't have to worry about, if they have aquaporins, and animals do, the cells don't have to worry about either expanding and exploding or shrinking and collapsing, because water is tending to come out versus go in at the same rate. So osmoconformers don't need to worry about water balance. That's most marine animals. What about freshwater animals? Can they be osmoconformers? Okay, no way. If you're a freshwater animal, you can't be an osmoconformer because your solute concentrations would be too low. So osmoregulators so they have a different body fluid total solute concentration than the environment. So that's so most marine fish, although sharks are osmoconformers, but most marine fish are osmoregulators for a reason I'll mention in a moment. Oops. It's marine animals as osmoconformers, but marine fish as osmoregulators, so we'll come back to that. Okay, so most marine animals, which would be mostly inverts, right? Because most marine animals are invertebrates, right? Okay, makes sense? Okay, so sharks also are osmoconformers. Marine teleost fish, most marine fish, are osmoregulators. There's a reason for that. Teleost fish left, the ancestors of teleost fish left marine environments, moved into freshwater environments. And when they left marine environments, it appears that salt concentrations in Seawater were somewhere in the range of 300, 400, 500 milliosmoles. Right now, they're around 1,000. Today, ocean, and I'm rounding. The ocean is about 1,000 milliosmoles. Fresh waters, anywhere from near zero to maybe 30, 40, 50 milliosmoles. Above that, you start calling it brackish, partly salt, okay? So teleosts are around 300 to 500 milliosmoles. One's living in fresh water, typically at the low end. Those in marine environments, typically at the higher end. What about us humans? We're at around 300 milliosmoles. A little under that, but close. Okay. Michael? So all fish are, all teleost fish are osmoregulators, whether they live in marine or freshwater environments. Okay. And an osmoregulator is an animal that has a different solute concentration in its body than in the environment. Okay. Okay, so why are teleost fish different? Well, teleost fish, the ancestors of modern marine teleost fish, were freshwater at a time when ocean concentrations were somewhere in the neighborhood of 300, 400, 500 milliosmoles. Okay. And so they were adapted, presumably these ancestors were adapted as osmoconformers to those salt concentrations. Moving into freshwater environments, of course, now they have to maintain a different solute concentration. And so they maintain solute concentrations that are a little lower than that. It's energetically a little cheaper to be on the low end, so they have evolved to tolerate lower body solute concentrations. Okay. And those are also the ancestors of terrestrial animals. So terrestrial vertebrates tend to have body fluid compositions of somewhere around 300 milliosmoles. Modern marine teleosts moved back into the oceans, but by the time they moved back, the ocean was different enough in solute concentration that they couldn't tolerate it as osmoconformers. So they were stuck being osmoregulators, and they still have to do that. So it's energetically more expensive, but they're stuck. They couldn't go in one massive change to becoming osmoconformers again. Does that make sense? Okay. So you could have selection, right? You could have selection on proteins to tolerate different concentrations. You'd have to switch some amino acids around, but you can have selection to tolerate different concentration of salts, but that same protein now, if you lower the salt concentration, now it's going to be denatured because it doesn't have the ions that are helping it hold its shape. Okay. Okay, so what do osmoregulators do? So if you're an osmoconformer, you can have an outer epithelial layer. So here's an epithelial layer with a basement membrane and cells underneath with some extracellular matrix. And if the concentrations are the same, solute concentrations are exactly the same, then water moving in and out through aquaporins there'll be no net movement. The concentrations are the same. Water coming in will be balanced by water going out. No problem. So that's an osmoconformer. I'm going to draw these cells a bit bigger, so if you're sketching along, you might want to draw them a bit bigger, because we're going to need to put some stuff inside them. Again, tight junctions here. Again, that's an epithelial layer. Again, there's a basement membrane. So here's a marine fish, 1,000 milliosmoles outside, 300 milliosmoles inside. What's water going to do? Okay, water is going to tend to leave. So that means no aquaporins over most of its body. Okay, no aquaporins. Every time it swallows a meal, what's going to happen? What's it taking in along with its food? Seawater with too many ions, right? Too many sodiums. So what do? They have in their gills in particular sodium-potassium pumps, pumping out sodiums. And, of course, they continue cycling potassiums, but they're going to have a net movement of cations out of the body, right? Pump out sodium, allow potassiums to follow. And then chloride will follow passively. Make sense? Okay, if you keep moving positive ions out, the inside gets more negative. It's going to repel negative charge. Chlorides are going to be repelled. They're going to leave. Okay, so that's what's helping keep the solute concentration low. So the fish are swallowing water because they're always losing some. Wherever they have a gap, wherever they happen to have an aquaporin or some kind of gap, they're always losing water. So they're swallowing water and excreting the ions, if you're a marine fish. If you're a freshwater fish, you're tending to lose ions, and so freshwater fish are constantly pumping ions in the opposite direction. With their gills, they're pumping ions into the body. Okay, and they're excreting a very dilute urine. They're getting rid of water. We'll talk about the excretion in a moment. Okay, take a moment now to go through the problem. What is the problem of water balance and solute balance? Why do you need the right proportion of ions? Picture proteins denaturing, if they have too many or too few sodiums, chlorides, potassiums, and so on, or ureas or ammonium. So close your eyes and run through that a few times. And do the same thing for an osmoconformer, gaining and losing water, no problem, right? Same solute concentrations. If you're marine, it's around 1,000 milliosmoles. And then think about a fish, more complex. Fish is always taking in excess salt, has to get rid of it. Okay, I've shown you the simplest version, getting salt across the gills. Marine fish do that. They also get rid of salt in their kidneys. Okay. Okay, any questions? Yeah, Gil.
Sorry, what's going on with the aquaporins and the aquaconformer?
Sorry, with the...
What's going on with the aquaporins and the aquaconformer?
So in an osmoconformer, they can have aquaporins anywhere because the solute concentrations are the same in and outside of cells, extracellular fluid, and external environment. Okay, and they don't have to worry about excess water either exploding a cell or shrinking a cell. osmoregulators, they do have to adjust where they put their aquaporins, where they allow water in and out. Okay? So one of the things tight junctions and an epithelial barrier does is if they're complete, if the tight junctions are complete, you get a waterproof seal, and if there are no aquaporins. Okay. Jonathan? Yep.
You said that in the freshwater osmoregulators, the
Right, so in freshwater, it's the reverse problem, right? So if you're in freshwater, the outside is down at, say, 30 milliosmoles. And so now the problem is you're constantly swallowing water that has low solutes, so you don't have enough sodium, chloride, potassium, and so on. So if you're freshwater, instead of having abundant sodium potassium pumps on the outside of the cells, pumping them out, okay, which is what green teleosts are doing, you put the sodium potassium pumps and channels on the inside of the basement of the epithelial layer, the basal side, and you pump sodium and chloride into the cell. And we won't talk about all the mechanisms, but with some additional pumps and channels, they can actually gain salts from the environment, even that low solute environment, and move them in. So I talk more about that in my physiology class. But so they're constantly moving sodium and potassium and chloride into the body instead of out. Okay. Destiny?
So just to clarify, so the green one on the left is the conformer, and then this black one on the right is the regulator?
Yep. Okay? Okay, so we've talked about ammonia and ammonium ion. We've talked about urea. Talked about ways those are being moved. Uric acid, we won't talk about in its structure in more detail, but it is low, has low solubility, it crystallizes easily if the amount of water decreases. So if you have a solution of uric acid, so imagine we had uric acid, you guys are the inside of the tubule of a kidney, okay, of any kind of excretory organ, okay, and there are uric acid molecules dissolved. If we moved some water out, okay, then the concentration of uric acid would be higher. Let's say you guys are constantly using sodium cotransporters to move sodium and glucose and sodium and amino acids back out into the tissues of the body. Here's the lumen up above us, okay? And chloride would follow, right? If you're moving sodium out and you had chloride channels, chloride would follow to balance the charge. You're moving all these solutes out. If you have aquaporins in our cells here, what happens to the water? Does it stay in the lumen? Okay. Water is going to follow. Does that make sense? Okay. So water is following. What happens to the uric acid? Does it stay the same concentration above us? So the uric acid becomes more concentrated. It's not very soluble, so it starts to crystallize. Okay. That's a useful thing if you're trying to get rid of uric acid, because any two that are crystallized, any two that are next to each other, are no longer two solute particles. They're now one solute particle. And as the crystal grows, you have fewer and fewer solute particles there. Okay, we're going to come back to why that matters. That's a useful thing if it happens in the right locations. If it happens in the wrong locations, it causes gout, which is crystals forming in your joints. Crystals of uric acid can be other things. Forming in your joints, very painful, not a good thing. It's like putting sand in your joints. Okay, so we're going to come back to that. But I want you thinking a little bit more generally about different excretory organs. Remember, we've talked about protonephridia, got a flame cell, there are slits, and so we get water, amino acids, glucose, wastes, ions, and so on. We recover glucose, amino acids, sodium, chloride, right? Secondary active transport. Okay, we recover those. And the wastes are excreted. In a metanephridium, it's just a tube with an opening. But we have a closed circulatory system. So all the proteins stay in the blood vessels. So outside of the blood vessels, the extracellular fluid, all we have are small molecules and water. Okay? So that's what gets into the metanephridium. Again, we use secondary active transport and wastes are excreted. Okay? I want you to take a moment to appreciate the beauty and elegance of this system. There are an incredible array of toxic molecules in the environment. Other animals keep inventing them, partly to use as toxins. Humans invent them. We've invented something like 80,000 different chemicals that never existed on Earth before us, many of which are toxic. And yet, excretory systems can get rid of them. Okay? Because as long as they're small molecules, and that is a key point, as long as they're small enough, they'll get into the excretory tubule, and they're just never recovered. So valuable molecules are recovered, water can be recovered, but the waste molecules just stay, and out they go every time you pee. Okay, it's an incredibly beautiful, elegant system, and when you see the nephron, which we're going to start going through next, it's just an astonishingly beautiful part of a wonderful organ. You're going to love the beauty of the kidney. Okay, green glands. So there are a whole lot of ways to do excretory organs. They all do some variation on these themes using many of the same molecules. Again, small gaps. So you get water So you get water, sodium, chloride, amino acids, glucose, wastes, all get in. Proteins don't. So crustaceans have open circulatory systems. They have transport proteins, including oxygen-binding proteins, in their hemolymph. And this term is going to matter. So if you have an open circulatory system, your lymph is your blood. So we call it hemolymph. If you have a closed circulatory system like we do, you have blood in the circulatory system, lymph as extracellular fluid. Okay? Hemolymph when they're the same thing. So the proteins don't get in. And of course, neither do cells. Again, you use secondary active transporters to recover glucose, amino acids. Chloride follows passively. And what you lose are wastes. Okay? So if I gave you some unknown phylum, could be an exam question, and said, predict how its circulatory system works, and if you knew whether it was an open or a closed circulatory system, you could make some really good predictions about how it works. Okay? Details might be wrong, but your general predictions would be right. A couple terms to remember. Filtration. Filtration means let the small molecules in, keep out the big ones. Okay, keep out the proteins. So when we talk about filtration in the context of excretion, that's what we mean. Usually things the size of amino acids, glucose, most waste molecules, they fit. Okay, bigger things like proteins don't. Reabsorption, you get the valuable molecules back. Same way you do same transporters as you have in your digestive system. You're getting the valuable small molecules back in the same way. Okay, so reabsorption. And in the case of the green gland of crustaceans, it looks sort of greenish, hence the name, at least in some species. These two processes are happening just like we've been talking about. And you could propose a structure like the one I sketched up there for a green gland. Okay, one more. Yeah, Meg.
For a system like this, there's some molecules that I know are not efficiently filtered out, like in non-persistent.
Right. So, yeah, so why are some things not filtered out very efficiently? Mercury is one that we tend to hang on to. The problem with mercury is that it can substitute for anything else, any metal with a similar charge. Okay. So it is substituting for metals in the body and getting trapped. Any bound mercury gets trapped. That can be as part of proteins. So some proteins have metal cofactors. We're going to talk about hemoglobin, which does that, iron in hemoglobin. Proteins with a metal cofactor can wind up with a magnesium in there. Magnesium can substitute for a sulfur. There are lots of cysteine-cysteine disulfide bridges. So magnesium can substitute for other ions. And when it does, the proteins have a different conformation, and they don't function properly. And the problem is a lot of the mercury you take in is that. It gets stuck, and you can't get rid of it. You do excrete mercury or any other metal, but it's not fast enough. And it's because those charged metals wind up interacting very effectively with proteins, and they get trapped. Okay? And that's the problems with some toxins. Non-water-soluble toxins, they tend to build up. So we build up things like PCBs. Because they're not very water-soluble, they tend to get associated with fats and lipids, and you hang on to them much longer. Okay, Malpighian tubules, it's a little different, and I want to talk about it more because of the way they conserve water. But Malpighian tubules, they work by having gaps between cells, and they actively pump potassium into the lumen. Chloride follows passively, and that means we now have a higher solute concentration inside the lumen than outside. So what's water going to do? Water is going to tend to follow. And that brings with it wastes and some valuable molecules. Now, the valuable molecules are recovered just like we talked about. The wastes include uric acid. But it's very dilute. So it's still dissolved. Okay? Make sense? Okay, so if we looked at the structure, here's food. Here's the Malpighian tubule on both sides. Okay? And so what we've got is uric acid moving in okay, so uric acid still dilute. Now, these cells are reabsorbing amino acids, glucose, sodium, ions. And so what happens to the uric acid? Does that make sense? Take the water out, and the uric acid starts to crystallize. So we get more and more and bigger and insects, it's not always pasty white, but insects, if it's high in uric acid, you get this kind of whitish feces. Okay? If it has a lot of other stuff, it can be other colors. Birds do the same thing. In birds, the urine that develops in the kidney empties into the digestive system. And uric acid crystals form in the last part of the digestive system, and so they're excreting this very dry, uric acid-rich feces. So it allows them to the thing about using uric acid is as you take the water out and it crystallizes, you haven't changed the total concentration of solutes. And so you can just keep taking more and more water out. The crystals grow bigger and bigger. Okay? But you're not fighting a higher and higher concentration. The actual dissolved concentration of fluids in this excretory material isn't changing much because uric acid keeps crystallizing. Okay? So they can do this very efficient mechanism to produce really dry feces. Birds, reptiles, insects do it. Mammals don't. It's a problem for mammals, and so we have more trouble conserving water than these other animals do. If we did it, you would have uric acid crystals forming in your bladder, or even in the kidney tubules, kidney stones, or you can block bladder. Really painful stuff, not a good thing. Okay, so we use urea, which takes more water to get rid of the uric acid. So water follows, right? So when you conserve these other molecules, water follows. Okay, I hoped to get started on mammalian kidney, but we've already talked about a lot of the principles, so we'll come back to that and talk a bit about homeostasis on Friday. A's through N's, I think, are down here. O's through Z at the back of the room. The TAs are going to have them. If you don't have to rush to another class, take your time to come down.